All species of sea turtle within U.S. waters are listed under the Endangered Species Act. NOAA partners with U.S. Fish and Wildlife Service to implement recovery plans. NOAA's role is primarily to focus on recovery actions in the ocean. Sea turtles are facing a number of threats. Historically, all sea turtles were harvested for their eggs and meat. Once those populations were starting to decline because of the overharvest, then you got destruction of nesting beaches. The big one preventing their recovery is fisheries bycatch.
For this research, we sequenced the genomes of green sea turtles and leatherback sea turtles. They're representative of the two living sea turtle families.
We chose these two families because they are very different in their morphology, in their habitats, in their ecology. They have the DNA that we need in their blood, so we can just take a blood sample and then use this to get high quality DNA.
For the leatherbacks, we were able to collect fresh samples.
We use this ultra high molecular weight DNA and sequence it using short read technologies, long read technologies, and some other newer technologies to build these really high quality reference genomes.
The different technologies that have advanced in the last 5+ years have just completely revolutionized what we can do with genomes.
We ended up with a fully annotated reference genome that really gives us something to map everything to moving forward.
Essentially what we found is that despite green and leatherback turtles evolving from one another a long, long time ago, their genomes are actually very similar. Once we saw this, we started to look for differences. So what makes a green turtle unique and what makes a leatherback turtle unique?
We found a higher number of genes involved with olfaction and immunity in the green turtle compared to the leatherback. So this suggests that green turtles have both a better sense of smell than a leatherback and also have a better immune system. And we think the drivers of this are related to their habitat. So leatherback turtles exist in these pelagic environments, open water, cold, whereas green turtles inhabit these near-shore coastal environments that are a lot warmer. So there's a higher prevalence of pathogens in warmer waters. Sea turtle populations as a whole have generally declined over the last 50 years or so.
One of the main existential threats that sea turtles are facing is climate change.
We wanted to use these genomes to be able to analyze population level trends and see if there is information on how these species will be able to adapt in the future.
We know that genetic diversity is really important in terms of having the ability to adapt to different environmental conditions. When we looked at the green turtle compared to leatherback turtle, we saw that the leatherback turtle has very low genetic diversity. That gives us pause in terms of their ability to adapt to environmental change. There's a lot of changes happening now at an extremely rapid pace, and so ultimately their ability to adapt to these new conditions will be partly determined on their genetic variability. We're facing a crisis of biodiversity loss, but if we understand more about their evolutionary histories, we can make smart decisions for their recoveries.
This is really just the first step in advancing our understanding and implementation for conservation. These provide a really high quality resource for researchers in the future.
One really fundamental way that we're going to have to rethink what NOAA does in terms of recovery is implications for adaptation. We're now able to look at genes and better understand the potential for these species to adapt. That's really important with climate change in particular. We can actually start to focus on genes themselves and reassess populations. This could be a game changer if we can identify genes that are really important to survival. It's an exciting new era.